Okay, so let's just, dive right in. For decades, when scientists looked at a tissue, say a tumor or a piece of the lung, they were essentially looking at a blended smoothie.
Exactly. A smoothie, you just saw the average of millions of cells all mixed together.
But now, with single-cell sequencing, or scRNA-seq, we have this incredibly high-definition picture. We can see the whole bustling city where every single cell has its own unique job.
It's a huge leap. It's the difference between seeing a crowd and knowing what every single person in that crowd is doing. And that ability to see the heterogeneity is what's driving all these new discoveries.
Especially in something like cancer, right, or the immune system.
Absolutely. But, with all that amazing data comes a massive headache.
The complexity. You get data biases, and you also get these things called batch effects.
Yeah, batch effects. It's basically just technical noise. I mean, you run one sample in a lab in Boston on a Tuesday and another in a lab in London on a Friday.
And even if it's the same biological sample, the data looks different just because the machines or the conditions were slightly different.
Precisely. The data is noisy. So the field turned to a pretty revolutionary idea from the world of AI. They borrowed the thinking behind large language models.
Your GPTs, your BERTs.
Right. And they applied it to biology.
Uh-oh.
And that gave us what we call single-cell LLMs, or CCLMs, models like CBER, Genoformer, and scGPT.
And this is where the analogy, I think, really clicks. For you listening, these models treat the entire profile of a single cell, all of its genes, and how much they're expressed.
They treat that as a single sentence.
And each gene is a word in that sentence.
Exactly. And they learn this biological language by training on just enormous data sets. We're talking 30, 40, 50 million healthy human cells. They learn to predict a missing gene based on the context of all the other genes around it.
So they learn the fundamental grammar of biology.
They do. They build this incredible foundation of knowledge.
Which sounds amazing, like problem solved. But our deep dive today is all about a new framework called scPEFT. And it was designed to fix two really critical, almost paralyzing problems that pop up the second these models meet a real-world disease.
And those two problems are, first, performing poorly in what we call out-of-context scenarios, and second, this really scary thing called catastrophic forgetting.
Okay, let's start with the context problem. If these models have learned the universal rules of biology, why do they suddenly fail when they see a new disease?
Well, because that massive training data, it's mostly based on normal cells. So the model knows the universal dictionary of health, but then you show it something it's never seen before.
Something totally out of context.
Right, like the chaotic environment of a lung cancer tumor, or the immune system's response to COVID-19, or even just trying to apply human data to, say, a mouse. The model just gets confused.
Okay, so the standard AI solution to that is to fine-tune it. You take your big general model and you retrain all of its parameters on the new specific disease data. But that's where the second problem, catastrophic forgetting, comes in.
It's this really painful trade-off. Let's use an analogy. Imagine your foundation model is a perfectly tuned grand piano. It has learned all the rules of classical music.
It knows the fundamentals. It can play anything from that rule book.
Yes. But now you need it to play one very specific, very niche jazz riff.
So with full fine-tuning, I'm essentially retuning every single string and hammer on that entire piano just for that one jazz riff.
That's exactly what you're doing. And it gets really good at that one riff, but then you ask it to go back and play the original concerto.
And it's forgotten how. The fundamental knowledge, the core rules, have been overwritten.
They're just gone. And this isn't just theoretical. The research shows it happening. When they fully fine-tuned scGPT on disease data, its error rate for identifying basic CD4 proliferating cells, a really fundamental cell type, it doubled, jumped from 6% to 13%.
Wow. So it literally forgot what a common immune cell looks like. In a clinical setting, that's a disaster.
A total disaster. And on top of that, full fine-tuning is just incredibly expensive. It takes huge amounts of data and a ton of GPU power that smaller labs just don't have.
Okay, so let's just recap where we are about 5 minutes in. We've got these incredible single-cell language models that know the language of life, but they get confused by new diseases. And the old fix, full fine-tuning, makes them forget what they originally learned and costs a fortune.
So we're stuck. We need a way to surgically tweak the model without giving it amnesia.
Which brings up a good question for you to think about. Why is it so critical to preserve that original biological knowledge? Why can't we just specialize?
I think it's because disease isn't a new language. It's a deviation from the original one. The rules for how a cell divides or makes a protein are mostly the same in health and in cancer. You need the baseline to see the deviation.
If the model forgets what a normal T cell is, it has zero chance of spotting a slightly different cancerous one.
Exactly. You lose your anchor.
All right, so let's talk about the surgical solution, this framework. CPEFT. How does it work? How do we tune the piano without breaking it?
So CPEFT uses these small, pluggable software modules called adapters. And here's the absolute key. The original, huge foundation model. All of its parameters are completely frozen. You don't touch them.
You lock it down. So the core knowledge base is safe. And the adapters, their job is just to provide a tiny, focused change. A delta for the new context.
Precisely. And since you're only training the tiny number of parameters inside these little adapters, you prevent the catastrophic forgetting, and you save a massive amount of computational power.
The paper outlines 4 different types of adapters, like 4 specialized tools.
Yeah, you could think of it like a toolkit. Each one solves a different problem. The first one is the token adapter.
And this one tackles that batch effect problem we talked about earlier, the noise.
It does. It sits right at the beginning, where the genes, the words, enter the system. It's basically a small autoencoder that cleans up the gene signal, filtering out the technical noise from different labs or machines. It makes sure the model sees a clean word no matter where it came from.
Okay, so that cleans the input. Next up is the prefix adapter. This sounds like you're giving the model a little hint before it starts reading.
That's a perfect analogy. The prefix adapter literally adds a few tunable bits of information, like a little sticky note, to the beginning of the gene sequence. It's a way of telling the model, hey, pay attention, we're looking at COVID-19 data now. It injects that task-specific context right at the start.
And then there's LoRA, which is pretty famous in the AI world. Low-rank adaptation. What's its job inside the model?
So LoRa's job is to subtly shift the model's attention. The transformer needs to figure out which genes are most related to each other. Full fine-tuning would retrain these massive attention matrices. LoRa is much smarter.
It doesn't touch the big matrix.
No. It just adds 2 very small trainable matrices on top of it. This allows it to approximate the big adjustments needed for the new disease, but in a very efficient way. And crucially, these adjustments start at 0, so there's no distortion at the beginning.
Clever. Okay, and the last tool in the kit is the encoder adapter.
Correct. This one comes in a bit later in the process. After the model has figured out the gene relationships, the encoder adapter helps customize the final meaning, the overall context of the sentence for the new biological situation. It makes sure the new knowledge is integrated properly.
The numbers and the efficiency here are they're staggering. How much smaller is this toolkit compared to retraining the whole model?
We're talking about training only 0.05% to about 4% of the total parameters. It's tiny.
And what does that mean for resources? For a lab trying to use this?
It means you're using less than half the GPU memory. Suddenly, this kind of high-end AI becomes accessible to way more researchers. And for, say, a clinical application, the extra processing time is tiny, about 0.2 milliseconds per sample. It's basically instant.
Okay, let's do another quick recap. At the 10-minute mark, what we've learned is CPEFT uses this set of 4 small plug-and-play adapters token, prefix, LoRA, and encoder to surgically calibrate a huge biological AI. It freezes the original model to prevent it from forgetting everything, and the resource savings are just enormous.
It's a really elegant solution.
But it raises the obvious question. We're using a tiny fraction of the parameters. We're often using smaller, more specialized data sets to train these adapters. Do we take a hit on performance? I mean, does the accuracy actually hold up?
That is the million-dollar question, and the evidence is pretty clear. Not only does it hold up, it actually improves on the old method dramatically.
So let's look at the results. What happens when you use CPEFT on complex diseases? Non-small cell lung cancer, MS, COVID-19.
In those cases, CPEFT delivered accuracy improvements between, get this, 39% and 82% compared to the original out-of-the-box models.
Wow. Okay, but how did it compare to the fully fine-tuned models, the ones that were retrained completely?
Even there, it was better. It gave between a 4% and 15% improvement over the fine-tuned models.
Because it didn't forget the basics. It kept that foundation of biological knowledge that the other models just threw away.
Exactly. And you see that most clearly when you look for rare cell types. The fine-tuned model completely missed certain rare immune cells, like XCL11 cells. But CPEFT, because it had the broader context, it spotted them.
And it was also more robust when you had less data, right?
Much more robust. When they reduced the amount of training data, the performance of the fully fine-tuned model just cratered. It fell off a cliff. But CPEFT's performance stayed pretty stable. Fewer parameters means you need less data to get them right.
So it's not just about labeling cells correctly. The paper says CPEFT gives us new biological insights by looking at the model's attention scores.
Right, this is so cool. It's like looking inside the AI's brain. You can see which genes the model is focusing on to make a decision.
What's an example of that?
So in the COVID-19 T-cell data, CPEFT highlighted some known immune genes, which is good, a nice sanity check. But it also uniquely focused on two other genes, CEBPD and SCART1.
And what do they do?
Well, they're involved in how T-cells become killers and how they find their way to infected tissues. This is the model pointing to a specific biological mechanism relevant to this specific disease. It's a discovery tool.
That's incredible. It's moving beyond just classification to actual biological discovery.
It is. And we can push that idea of universal knowledge even further. I mean, think about the ultimate test.
Yeah.
Crossing the species divide.
Right. It's totally impractical to build a new one of these massive models for every single research animal, a mouse model, a macaque model.
You want to be able to leverage that huge human data set. So they tried adapting the human-trained scGPT to mouse data.
How did it do?
The native human model is okay, about 75% accurate on mouse cells. But with CPEFT, that performance shot up significantly. But the really wild experiment was adapting the human model to a nematode worm, C. elegans.
A worm? I mean, that's a huge evolutionary jump. The biology should be completely different.
Millions and millions of years of separation. And the native human model, as you'd expect, really struggled. But with CPEFT, it achieved nearly 80% accuracy in classifying the worm's cells.
80%? That's astounding.
It's a 145% improvement. It proves that there's some fundamental biological grammar that CPEFT can tap into and transfer across vast evolutionary distances.
So it can work with known cell types, even across species. What about finding new things, unsupervised discovery?
You can do that too. In some bone marrow data, without any prior labels, the model identified a unique subgroup of mature B cells.
What was special about them?
Their gene profiles showed they still had remnants of their developmental programs. They looked more like progenitor cells than fully mature ones, something standard markers had completely missed.
So the AI saw a pattern, a structure that human-led analysis had just smoothed over.
Exactly. And when they dug into those new clusters, they found they were functionally distinct. One was enriched for genes involved in antigen presentation, suggesting it was on a whole different differentiation path. CPEFT literally revealed hidden biology.
This really feels like a definitive step forward. CPEFT seems to be this modular, incredibly efficient framework that just solves the biggest problems holding back these foundational models.
It solves catastrophic forgetting, and it fixes the out-of-context problem. It just lets these models be useful across diseases, across species, in almost any scenario.
And the method itself is built to grow, right? You could integrate other data types, like from proteomics or imaging.
For sure. The framework is extensible. You can imagine a future where you combine all these different data types for a truly complete picture of a single cell's state.
So let's just end with a final thought here. If we can train an AI on millions of human cells and then efficiently adapt all of that knowledge to spot tiny differences in cancer cells or even to accurately map the cells of a worm, what does that tell us? What fundamental conserved biological grammar has scPEFT started to uncover?
It suggests there's a truly universal language of life encoded in our genes, one that's far more consistent and transferable than we ever thought. And so the real question for you to think about is this. If we can now leverage that universal language to pinpoint disease with this kind of precision and efficiency, how is that going to change the way we design personalized medicine? That power is finally within reach.